Good afternoon. This is chapter 13 out of your Martini Human Anatomy book. This is covering the nervous system and specifically nervous tissue in this chapter. Introduction here. So the nervous system characteristics. The thing we want to remember the most is the brain and spinal cord is going to control and coordinate every single aspect of the body. It's going to control and adjust every single activity of the body and provide swift but brief responses. Your nervous system consists of all the nervous tissue and includes our central nervous system, which is our brain and our spinal cord. Sometimes you'll read literature talking about the nuances of certain cranial nerves, specifically cranial nerve 1 and 2 being part of the central nervous system. Some are going to classify it as peripheral, but that's okay. We're just going to talk about central nervous system as brain and spinal cord. The peripheral nervous system, this side over here, is everything else. Couple of cool pictures here from the body's exhibit. You can see brain and eyes and spinal cord and some of the nerves. Same thing here. We could see the brain, the spinal cord, and some of our nerves. And then this cool picture over here is a microscopic view of the nerves. Another couple of cool views here of the nervous system to keep in mind as we are looking at all of the different nerves and components. There's these different divisions that we like to classify how we're teaching this and studying this. So in general, we see the nervous system at the top. Over here, we have the central nervous system, the brain, and the spinal cord. Over here, we have what we consider the peripheral nervous system. In this peripheral nervous system, we're going to classify things out into somatonervous system and autonomic nervous system. Yes, there are components of the central nervous system, obviously, that are going to coordinate autonomic nervous system. This is just a way that they like to separate these things out. So our autonomic nervous system, or the automatic, is the fight or flight or sympathetic division, and the parasympathetic or rest and digest. And please note, again, there is a lot of component here with the central nervous system as well. And this is just how Marib likes to classify things. And then over here, we look at these terms, somatonervous system. And this is really going to be like your muscles and your bones, or like what you would draw if you were drawing the outside of the body. And then we look here and we see sensory, how we feel things, or the term afferent will go along with the word sensory. Okay. And then motor or efferent will go along with the word motor. So we see a sensory component, how we're feeling things, and then we see a motor component, how we're moving our bodies. So brain and spinal cord, central nervous system, responsible for integrating, processing, and coordinating everything that we feel and all of our movements. It is also the seat of intelligence, memory, learning, and emotion. Some terminology again. Sensory afferent. I like to remember this as SAD. Sensory afferent, and then dorsal will come up later. The dorsal aspect of our spinal cord is going to be the sensory afferent component. So this means that it's just bringing information from our body that is interacting with the world, right? So think about the sensory receptors in your skin, and then brings that information towards your central nervous system or your brain and spinal cord. So this is just how we feel things. This can be further subdivided into somato and visceral. We'll look at somato and visceral on the next slide. Efferent is the motor, how we move. Okay, so this is going to be commands coming from our brain and our spinal cord. So our somato portion here, somato portion, this is just monitoring skeletal muscles and joints. Visceral portion is monitoring smooth muscles, heart muscle, and your other internal organs. So when you see visceral, just think internal organs. Efferent, or the motor component, our somatonerves regulates our skeletal muscle contractions. Our autonomic nerves is that's autonomic is kind of like the word automatic. Okay. Regulates internal organ activities without us even having to think about it. Okay. Our heart muscle contractions, smooth muscle contractions, our glands. We can see this basic component of a neuron structure here. And so we see the dendrites here, these little branches, okay, are the dendrites. And we'll see another picture of this as well. Okay, so we see these little dendrites. They are stimulated by environmental changes or the activities of other cells. And then this is the cell body in here. And this would be the cell body. And then you can see all the little organelles here. Okay. Well, dendritic spines wouldn't be organelles. There you go. And then coming through here, let's go back in. You can see what we call the axon hillock. It's just this little funnel from the cell body to the axon. That axon is going to be the way that the electricity, or what we call the action potential, or this impulse, is going to travel. Okay? So think of the wiring that we see in our home, conducting electricity. So it's going this way. Okay. And then it's going to have these little terminal boutons or axon terminals, the end of the axon, and then that's going to connect to the next cell. Okay, so through electricity and chemistry. Okay, so chemicals that we call neurotransmitters and other chemicals as well. But basically we'll just focus in on neurotransmitters for the most part. But electricity and chemistry is how this nervous system is going to run. So these electrical synapses, impulses can be conveyed in any direction. Sequence of events would be the presynaptic membrane of one neuron, tightly bound to the postsynaptic membrane of another neuron. This binding permits the passage of ions from one neuron to the next. So what does that mean? So the synapse is this right here. So if these were little fingers and you wrapped your hand around, so take your right hand, wrap it around your left wrist. Okay, so this right here would be your fingers. Okay? This right here would be your wrist. Where these two things are connecting. Technically, there is space between it, right? Feels like there's no space between, but there is. We are separate, and they're separate entities, right? For lack of a better word. And so in this space here, that's the synapse, right? In between these things here. That's this, we call that a synapse. Okay, that's the space. So what is happening there, right? Is what we look at here. So the presynaptic membrane is that first part, little axon terminals. The postsynaptic membrane, okay, is where it's going next. Okay. Okay, so let's look at some cellular organization in our nervous tissue. So nervous tissue consists of two cell types. We see neurons and we see neuroglia. Neurons, we think of these more like the functional units of the nervous system. Okay, the functional units of the nervous system. The neuroglia, we think of these more as the structure. They both have structure, they both have function. And so we don't really want to make that a concrete statement, but that's usually how it's taught, is the neurons are more functional, neuroglial are more supportive. So the neurons, we have 100 billion of them, right? 100 billion of them. Get the right color for that one. 100 billion of them, right? In comparison, neuroglia have 10 times that 100 billion, okay? 10 times 100 billion. Okay, so our neurons are nerve cells that are responsible for the transfer and processing of information in the nervous system. They consist of a soma, or the cell body, the axon, the dendrite, the dendritic spine, the axon terminals, what we just looked at. And then the neuroglia are more supporting cells. They protect the neurons. Okay? So we look at this as the neuron. The neuroglia are the support cells. And you can see a cool picture over here of a neuron or the pyramidal nerve cell, and we'll look at those later, growing on the surface of an astrocyte or astrocytic neuroglial cell. Okay, so pretty cool looking. And we'll look at specifics of those in a little bit. So, overview of our nervous system here. We're going to look at peripheral nervous system, providing sensory information towards the central nervous system and carries motor commands away from the central nervous system. Again, we're going to divide it out into afferent being sensory, okay? Afferent being sensory, and efferent being motor. Okay, so our afferent division brings sensory information to the central nervous system and begins at the receptors. So this could be the pressure receptors in your skin. This could be the hot or cold receptors in your skin. This could be your sight. Okay. Efferent division carries motor commands to the muscles and the glands and ends at the effectors. Neurons come in a variety of sizes and shapes, but all are variations. So another picture here of that same neuron. So we see the dendrites out here. We see the cell body in the middle. We see the axon hillock, which is this little funnel into, let's go here, the axon. Okay. And then the little end of that axon, we call that the axon terminal or axon terminal boutons. Okay. And then please note the impulse direction is this way. So electricity and chemistry coming in, okay, hitting those dendrites, information at the cell body, and then cruising this way. Okay, and then connecting in with the next cell that might look like this, might look like a different cell. So this is the typical motor neuron that we just discussed, but this is what it actually looks like. So you can kind of see it, right? So you can see the little dendrites here. These would be little dendrites here. And you can see the little spikes or spines on the dendrites right here. Okay. And then if you look close, this would be the cell body, obviously. We could see that a little bit easier than everything else, but here in blue, that would be that axon hillock or that funnel in. And when you see that, then you can pick out the axon pretty easy after that. Okay, so our dendrites, cell body, axon hillock, axon. And take a moment here and pause and go through this. That will help as we move through. So take a moment, pause, and then finish this up and then start playing again, and we'll go into the next slide. We're going to go ahead and look at the cellular organization in the neural tissue or these neuroglial cells. So again, glial being glue or the structural units. We're going to look at all of these cells, these astrocytes, oligodendrocytes, microglia, ependymal cells, and then the neuroglial of the periphery. We're going to see satellite and Schwann. So in the central nervous system here, we see these astrocytes. And when we look at this, astro means star, like astrology or astronomy. Okay, so this means star. Okay, so they're star cells, CYT. Oops, not sure why it's doing that. CYT is cell. So these are star cells. They have a large number of cytoplasmic processes. They control the chemical content of the interstitial environment. They help maintain the blood-brain barrier of the brain. Now, what this means is blood does not come in contact with the cells of your brain or your brain cells, that would be dangerous. Okay? That's what happens when there's a stroke, where there's a brain bleed. What does come in contact is your cerebral spinal fluid. Okay, so this is part of that barrier protecting our brain from the blood and things that might be in the blood. Okay, so that cerebral spinal fluid is an ultrafiltrate fluid. Okay, so astrocytes helping to maintain that blood-brain barrier. They isolate the neurons from the general circulation, and then damaged astrocytes could cause that the central nervous system became susceptible to a drug that should not affect it. Okay, so damaged astrocytes will have implications on that blood-brain barrier, is all that that's saying. So we can see our astrocyte here. Okay, this is the most abundant astrocyte A, abundant A, most abundant, versatile, highly branched glial cell. It clings to the neurons. Look at how it's clinging here. Isn't that interesting? So it clings to the neuron, okay? And it also clings to the blood vessel. Here's an artery. Okay, so this capillary here is a blood vessel, and the artery feeds into that capillary bed. Okay, and so you could see it's this kind of cell in between. So basically, it's checking in on this nerve to see what it needs. And then it's going to get what it needs from the capillary. Capillary is going to deliver blood and nutrients. And that astrocyte is hanging on to both of these things. Kind of cool, right? So supports embrace, plays role in exchange between that capillary and the neurons, guides migration of young neurons, controls the chemical environment around the neurons, and responds to nerve impulses and neurotransmitters. So this is the electricity, this is the chemistry. It influences neuronal functioning and participates in information processing in our brains. So again, hanging onto the nerve, hanging onto the blood vessel, and coordinating between the two. This is a pretty cool looking picture here. Astrocytic glial cell from the central nervous system. It's a scanning electron microscope. Okay, kind of looks like outer space, or maybe if you're into Harry Potter, these look like little Dementors cruising around, right? So pretty neat looking picture, I think. Another cell would be the oligodendrocytes, and these are in the central nervous system. These are extremely important for the myelin sheath, and we'll talk about myelin more. Okay, so these are cytoplasmic extensions, contact the soma, that cell body, okay, or the axons. And the cytoplasmic extensions tie axons together in this myelin sheath. Okay, that myelin sheath is going to increase the rate of conduction of a nerve impulse. Okay, so it's going to insulate it so it's faster and more effective. Okay. Microglia are another cell type, and they're the smallest of the neuroglia cells. They are phagocytic. So phage, think of the word esophagus, that PHAG, esophagus is in there. So that's eating. Okay, so these are the eating cells. These are part of the immune system. So they protect the neuron from by removing waste and debris. They're often found in infected or injured tissue from the central nervous system. Your immune system and your nervous system are really intertwined. There are schools of thought that call it one system, that they don't even separate the systems out. And it's called PNI, or psychoneuroimmunology. Psych meaning mind, neuro being nerves, immunology being the immune system. And there are really, really great scientists, really influential scientists out there. One by the name of Candace Pert. She is deceased, but she was responsible for finding the concept of receptors, right, for our body. She found the opioid receptor. She was head of the brain biochemistry department at the National Institute of Health, and she coined this phrase, psychoneuroimmunology. and decided that they should be just one system. So you'll see proof of that concept all throughout the nervous system, that the immune system and nervous system are linked, and obviously the mind. Okay. So here's our oligodendrocytes. Oligo means scanty or few. Okay. And what we see here are scanty or few, right, of these branched cells. And their processes are those little arms here, and their little arms right here, they wrap around that axon of this nerve in the central nervous system. Okay, central nervous system. Okay. They form these insulating myelin sheaths, which is this thicker nerve tissue, and we'll take a more expanded look at that in a moment. Here is another cool picture of a scanning electron microscope view of an oligodendrocyte cell. And this oligodendrocyte is the orange that you see here. And it's attached to an axon nerve fiber in the light green. So see the light green here, and then the orange kind of surrounding it, insulating it. To me, it kind of looks like a tempura green bean, right? But nope, it is an oligodendrocyte and the uh, axon fiber underneath. And here is a drawing of the same thing. Okay, so you could see that myelin sheath wrapping around, and you could see those nerve fibers there. And this is from your book. And that oligodendrocyte forms a myelin sheath, forms these little myelin sheath gaps or these internodes. And these are these areas with mostly myelinated axons. That's called white matter in the nervous system. And then areas without the myelin is called gray matter. So gray matter is your cell bodies and your dendrites. That's how we talk about it. We're going to talk about white matter. We're going to talk about gray matter. We'll talk about this again later. And white matter is the myelin, okay? And then the gray matter is the cell bodies and the dendrites. Microglial cells, these are small ovoid, or they kind of resemble an oval, okay? Ovoid. With thorny processes that touch and monitor neurons, they migrate towards injured neurons. So think this about that kind of neuroimmunology kind of overlap, right? They transform to eat up or phagocytize microorganisms and neuronal debris. So again, this is the primary immune of the nervous system. So the microglial cells where neuro and immune system really overlap. And this is the cute little picture of the microglial cell. So you could see that this is the defensive or the immune cell here. So our ependymal cells are next on the list. These line the ventricles or the spaces in our brain. And these spaces, these ventricles, are filled with cerebrospinal fluid, which is that ultrafiltrate that bathes and nourishes our central nervous system. Okay, so it lines the central canal of the spinal cord, or this little hole, basically, for cerebrospinal fluid in the middle of our spinal cord. It monitors the cerebrospinal fluid, and it might be ciliated. Remember those little hair cells, right? And it's involved in that cerebrospinal fluid production. And here you go. So here's the little cilia, these little hair-like projections. And this kind of looks like hairy teeth, right? So these look like teeth, and those are little hairs there. But no, these are the ependymal cells that line our cerebrospinal fluid-filled cavities, or our ventricles, you could call it. Okay, so down here would be the brain or spinal cord tissue. And then these cells here are the ependymal cells with their little hair-like projections. And they are responsible for the making of our cerebrospinal fluid. And here's a cool picture of a scanning electron micrograph of the brain of our ependymal cells here. So you can see the ependymal cells in yellow, and the little hairs of those ependymal cells, the ciliary hairs, are in green. And it's kind of interesting looking, right? And a lot of times I get feedback that this looks like little cookies, cookie crumbs in Cookie Monster's fur, right? Sometimes we think it looks like a coral reef. Sometimes we think it looks like shag carpet with some crumbs in it. But my fave is the Cookie Monster with the little cookies stuck in his fur. But nope, it is a pendymal cell here, okay, making our cerebral spinal fluid for us. So what are those ventricles? You can see the ventricles here in blue. And so the ventricles here in blue, and that is our cerebral spinal fluid is here, conducting electricity and nourishing our brain, giving it nutrients and balancing electrolytes. And then right here in the middle is where you see the so you can see the central canal here. Let me clear the screen. I'm going to put it in red. So follow this line down. See this little hole? That's that central canal. Okay, so the central canal where the cerebral spinal fluid is. So it's around here and in here. So it is in here and around your brain as well. Okay, providing buoyancy and making the brain feel a little bit like lighter. Just like when we go into water, we feel lighter, yeah, and supported and protected. So our neuroglia of the periphery here, so our satellite cells. So you can see the little satellite here, and it kind of looks like a little satellite, doesn't it? So our satellite cell regulates the exchange of material between the cell body and the environment. And then our Schwann cells, sometimes you might see it called neurolemmacytes, but we'll call them Schwann cells. They form the myelin sheath in the peripheral nervous system. So the oligodendrocytes form the myelin sheath in the central nervous system, and the Schwann cells form it in the peripheral nervous system. Okay. So this is vital to regeneration of damaged peripheral nerve fibers. And then we look here at these myelin sheath gaps in between, right? The myelin. And these gaps are sites where axon collaterals can emerge. And I highlighted that to kind of hang on to that for later, because we'll talk about that at the end when we talk about how nerves repair. And here's the picture of it for you. And another little study slide for you to look at. In the periphery, we see the satellite cells and the Schwann cells that we just talked about. So satellites surrounding the neuron cell body in the ganglia and regulating oxygen, carbon dioxide, nutrients, neurotransmitter, the chemistry really around the neuron here. And then the Schwann cells surround all the axons in the periphery, and they myelinate those peripheral axons and participate in the repair process. And here is that repair process. Okay, so we're going to look at this. It's a 3-component here. Okay. And so we'll start right here with number 1. So number 1, a Schwann cell envelops the axon. You can see that yellow line is the axon of the nerve that we labeled earlier on. Okay. And you can see how it's kind of rotating around there. And then number 2, the Schwann cell then rotates around the axon, wrapping its plasma membrane loosely around it in successive layers. Number 3, the Schwann cell cytoplasm is forced from between the membranes. The tight membrane wrappings surrounding the axon form the myelin sheath. So you can see how it's really tight around the front here and then not so much around there. So you can see the Schwann cell cytoplasm kind of outer edge area. Okay, just insulating that axon. And then here's a real picture of it. So kind of neat looking. In the centerpiece here is that axon. And then you can see here on the outer area here, these concentric rings of that myelin sheath. The organization of the neurons in the central nervous system, peripheral nervous system are not random. Okay, organization in the central nervous system we see a collection of cell bodies in one area, creating the gray matter. Okay, what makes white matter white is the myelin. Okay, the gray matter is going to be the cell bodies and dendrites. Okay, bundles of axons in an area create the white matter. We're going to call these tracks or columns. Okay, and then organization organization in the peripheral nervous system, the cell bodies are found in the ganglia. Okay, so we see ganglia for the cell bodies in the peripheral nervous system. The cell bodies in the central nervous system, we call them nuclei. Okay. So ganglia, periphery, nuclei, central. Okay. And then we see the axons bundled together to form the nerves, spinal nerves, cranial nerves, in the periphery. So we could see this drawing here, helping to organize what we've gone through. Central nervous system, we see astrocyte, leukodendro, microglia, and ependymal. So astrocytes, we want to remember that they maintain the blood-brain barrier. They provide structural support, regulate ions, nutrients, and dissolved gas concentrations, absorb and recycle neurotransmitters, and form scar tissues after injury. Oligodendrocytes, they are going to myelinate the central nervous system. Okay, so myelination of the central nervous system. Microglia, right, remove cell debris, wastes, pathogens by eating. Okay, so phagocytosis. Ependymal cells, it's going to make our cerebrospinal fluid. Okay, line the ventricles and central canal, assist in producing, circulating, and monitoring cerebrospinal fluid. So our neurons or our neuronal classification, we're going to see these functional classifications of neurons in these three different groups. So we see sensory, motor, and interneurons. Okay, so sensory, motor, and interneurons. And you see a component over here of sensory would be when you check a reflex, right? Like a patellar reflex. We're going to see this sensory information coming in through the dorsal aspect of the spinal cord right through here. Okay. And then you see this quick little reflex with the motor component. Okay. And so you saw sensory and motor. Sometimes you'll see an interneuron. And we'll take a look at that in another area. So this one is going to look just like that other one where we see the big picture here, and that is going to be this. So we'll go through this slide by slide. Okay, so this is going to explain the regeneration of a nerve fiber in a peripheral nerve. So there's a nerve damage, and how are we going to repair the nerve in the periphery? Okay. So number one on that list is this. So the way to look at this is it's this little part right here. So you already drew this. These are going to be the dendrites up here, and then this is the cell body, axon hillock, axon, and then axon terminals here. Okay, so you already drew that, so you're familiar with this, and then we're going to see this shape here expanded here. So the axon fragments or it gets damaged. Okay, so we see the cut here. Okay, so we see this axon fragments. The cut axon ends seal themselves off. The axon transport is interrupted, causing the ends to swell. Without access to the cell body, the axon and its myelin sheath begin to disintegrate, distal to the injury. Degeneration of this distal end of the cut axon is called Wallerian degeneration, spreads down the axon. So it's kind of like if you had a plant root and it got cut right here, right? And the main plant roots were here, and this was a little offshoot. Everything this way would start to dry out and die out, okay? And then everything this way could potentially still just stay alive. That's all it's saying. So everything this way is okay, okay? But it can't do its job to convey here. So everything along this path here is going to dry out like you just cut a root of a tree, and this part here starts to die. That's all that means. Number two, Schwann cells. So that's who's myelinating in the periphery, remember? So our Schwann cells and macrophages clean out the dead axon distal to the injury. Now remember our distal versus proximal. Distal is going to be further away from the center of our body. Okay. Proximal is in proximity to. Okay, so closer in. So surviving Schwann cells engulf the myelin fragments and secrete chemicals that recruit macrophages. Macrophages are big eaters. Okay, big eaters. These guys right here. These little Pac-Mans are going to chew up that axon debris or quote unquote garbage, right? That was left behind from the damage. Okay? Now these macrophages are going to dispose of the debris and they're going to stimulate the Schwann cells to divide. Okay, so they release chemicals here that stimulate the Schwann cells to divide. So this is a pro-inflammatory state right here. And so it is part of the demolition and the remodel. Like if you were remodeling your home, there would be a demolition component to the existing structures that are hanging out here. And then there would be a remodeling component. We're in the demolition phase here. And remember, it's chemistry that is driving this. Number 3, axon filaments grow through a regeneration tube. Schwann cells release growth factors and express cell adhesion molecules that encourage axon growth. Schwann cells line up along the tube of remaining endoneurium, endo being within, remember, neuro being nerve, forming a regeneration tube that guides the regenerating axon sprouts. See how the cute little sprouts forming through here? Right? Across the gap to their original contacts. Number 4, the axon regenerates, a new myelin sheath forms. Look, we see that this is healing. The Schwann cells protect and support the regenerating axon, this yellow part here, and ultimately produce a new myelin sheath around it. Oops. Good. Okay, so important to note in that process. Okay, so there is a substance called VEGF, or vasoendothelial growth factor. Vaso means blood vessel, but that's just how this was named. So they know that VEGF is all over the body for blood vessel growth, for nerve growth, for bone growth when we break a bone. So VEGF is important for all sorts of things. We're talking about it here with this paper that's pretty old. This information's been around for a while. It's well known. So VEGF promotes neurogenesis. Now, genesis, think of the word generate. Okay, so it's generating nerve when we see that dysfunction. So it's one of the chemicals that is part of the demolition and remodeling phase here. Okay, so the remodel, the recycling, the repair. VEGF has trophic effects in the neurons and the glia of both central nervous system and peripheral nervous system. VEGF supports neuronal migration in the developing central nervous system. VEGF is essential for neuroprotection. Really important, right? Essential for neuroprotection in adults. And then VEGF might be of therapeutic value in the treatment of neural disorders. Now, what's really important to understand here is this paper now tells us NSAIDs, like ibuprofen is an NSAID. Okay, NSAIDs are known to block at VEGF. So we have to understand this. If our patients have a nerve injury and they're trying to repair, right? And a lot of times that'll be painful. And so they might want to take a pain medication and they might reach for something like ibuprofen, which is an NSAID, and that's going to block this VEGF and it's going to slow that healing process down. So we need to be careful with this, right? The treatment with different NSAIDs significantly reduced VEGF expression regardless of the cell line. So this is blood vessel, this is nerve, this is bone, right? they were doing it with this bone with osteoblasts here, but they said it didn't matter which cell line it was, and instead studied. So it didn't matter what the NSAID was, didn't matter what cell it was blocking VEGF expression. We know we need VEGF in order to do these things. Okay? So we need to be very careful with these NSAIDs. So for example, some damage to myelin sheath, multiple sclerosis is a good example of that. Okay, so myelin sheath gaps between our adjacent Schwann cells here and sites where axon collaterals can emerge. So remember when we highlighted that in yellow at the bottom of the screen a couple slides back, and I said, hang on to this. This is important, okay, in understanding the nervous system. So what happens here with multiple sclerosis? Okay, we see this healthy nerve example over here. Okay, so we see a healthy nerve example over here, and then we see a nerve over here affected by multiple sclerosis. Notice the damaged myelin here. Okay, the damaged myelin. Okay, damaged myelin here. So the idea is that the immune system is stripping the wire here, right, in order to expose the fibers here. So sometimes what you'll see in papers, and a very common theory to the cause of multiple sclerosis would be the viral theory of MS. So viruses tend to invade our nerves, right? So herpes viruses specifically like to invade the nerves. And all that immune system activity, okay, they might need to strip that myelin sheath off of that axon in order to get to the axon to try and repair it. Okay, so remember we're seeing the repair mechanism in the periphery here, okay, and this occurs in the central nervous system, but it's pretty similar. Okay? And so what we know is we're going to then have this exposed fiber. The other component of this we want to remember is this exposed area is where these axon collaterals, these little sprouting guys, these little filaments, okay, so that's where these axon collaterals can emerge. So it's not specifically those little filaments, but remember that little growth of those filaments, right? So what collaterals are going to look like are little offshoots here. And let me use something a little easier to look at. So it's going to create these little offshoots. Okay. And we can only have these little offshoots where the axon collaterals emerge from the little sites here of myelin sheath gaps. So you need this exposed in order for the little collaterals to come out and bypass the damaged areas. Okay. Sites where axon collaterals can emerge is only at those myelin sheath gaps. So the body is making more myelin sheath gaps when it starts destroying its own myelin, right? And then what we see on imaging of the brain, okay, so this would be the nose right here. This would be the cerebellum right here, okay, front, back. And then we see these little scars. So see the little arrows there? Those white spots aren't supposed to be there. So the white spots aren't supposed to be there. So that's the scarring of multiple sclerosis, evidence of multiple sclerosis on imaging. So one of the theories of what's happening, right, of multiple sclerosis, right, is going to be our white blood cells, our immune system attacks the neurons, but our immune system just doesn't attack things out of nowhere. Okay, so it's this autoimmune system concept, okay, but it doesn't just happen out of nowhere. Okay, so the immune system is smart and is trying to do something. Okay, so what it's doing is it is attacking this myelin sheath for what reason? Well, myelin sheath gaps between our adjacent Schwann cells are the sites where axon collaterals can emerge. They can't come out of the area where the myelin sheath is. It can only come out of the areas where it is not. So in order to repair, it is creating more areas like this. So it can get some collaterals or offshoots, okay, out to bypass the nerve part that is damaged. Okay? And so it starts destroying that. Okay? The immune system's like, hey, I got to strip this wire. So think of it like a lamp. Okay, so you come in and the lamp's not working, and somehow you determine that maybe something ate through the wire, a little rat ate through the wire, right? And if we didn't live in a throwaway culture to where we would just throw the lamp out and like maybe order one on Amazon, right? And have it show up. No, we want to repair it, right? So what do? We splice and we take the covering off of the wire first. We take the covering off of the wire first, okay? And then we repair the wire, right? We repair the wire and then we wrap it with electrical tape, and now we have a functioning lamp again. Okay, so one of the theories here is that the immune system is taking the covering, which is the myelin sheath, off of the axon, which would be the wire, in order to repair. Okay, and we know what's going to help with that repair is going to be that VEGF. It's neuroprotective, it helps things regrow, and they understand it a lot more in the periphery than they do the central. Okay, and MS is more central nervous system. Okay. So one of the things we want to remember with that is this. Okay. So if NSAIDs block VEGF, okay, steroidal anti-inflammatories are going to block it as well. Okay, NSAIDs block VEGF, steroidal anti-inflammatories are going to block it as well. And one of the very first medications that a patient will be offered with multiple sclerosis would be a steroid. So we have to be very careful in understanding if we are setting people up to heal, okay, or not. Okay, and what all these different mechanisms do. So we call that the risk versus reward scenario. Okay, so understanding the normal mechanisms of healing of the body. Okay, so interesting theories there on the horizon. So a little mnemonic here that I like for remembering what cells are producing myelin in the nervous system. So these are our myelin producers right here. Okay, so central nervous system, okay, oligodendrocytes. Peripheral nervous system, Schwann cells, and the mnemonic is COPS. Okay, central nervous system, oligo. Peripheral, Schwann. And this is a nice little picture from your book talking about the white matter or the superhighways for our brain and our brainstem, and all throughout our spinal cord here. Okay, so we can see peripheral nervous system over here. So what makes white matter white is the myelin, okay? And that's the bundles of myelin on those axons, okay? And then the gray matter are the collections of cell bodies and dendrites, okay? And when that is in the peripheral nervous system, we call that ganglia. When it's in the central nervous system, we call it nuclei, collections of cell bodies, okay? So central nervous system, gray matter organization, we see the neuronal or the neural cortex, outer layer. Cortex is the outer layer. Okay. Gray matter on the surface of the brain. And then the nuclei are these little gray spots throughout here. Those are collections of neuronal cell bodies in the interior of the central nervous system. Okay, and then here the centers, collections of neuron cell bodies in the central nervous system. Each center has specific processing functions. Higher centers are the most complex centers in the brain. And then our white matter organization here, we're going to call them tracks or columns. And so these are bundles of central nervous system axons sharing a common origin and destination. So the columns are several tracks forming an anatomically distinct mass. Tracks, these are the centers and pathways or this information superhighway of our spinal cord up into our brain. Okay, so centers and pathways connecting the brain with other organs and systems in the body. And then you could see some examples over here of ascending or sensory tracks and some descending tracks here. Okay, so ascending coming from the finger here, and having like maybe thermoreception or pain reception, touch, pain, temperature, things like that. And then coming through, this is the dorsal root ganglion, this little bump here, and then it's going to cross over in the spinal cord and cruise up to the other side of the brain or cortex. Okay, and then you see motor starting here, okay, and cruises down, crosses over, depending on what it is. A lot of times it's going to cross over at the medulla, and then we see movement from that, the effector, move the hand. Okay. The end. So we've got a lot more chapters coming on the nervous system. That's just the start of it. So we'll go over a lot of these concepts again. Okay, so if it's a little foggy, a little unclear, that is okay for where we are right now. It will become clearer and clearer as we move. So this is a functional MRI, okay, of what we were just looking at here. Okay, so the sensory and motor components, but this is real. Okay, it looks like a beautiful piece of artwork, but it is real. So this is the front of the body here. This is the back of the body or the back of the head right there. And then we see these tracks that we were just talking about. Okay, so corticospinal tracks from the cortex to the spine. Okay, and we see all of this different information, superhighway pathways of the energy spraying around in the brain in very predictable patterns. So they know all of these different patterns here. So this is not just random, okay, firing of electricity and energy throughout the brain. But that's a real picture. So it's pretty cool looking. All right, here are citations of some of the books that I took it from. Obviously, I used your Martini book, and I added in some information from your Pearson book, and then a couple more neuro books here for you. There you go. I will see you for the next chapter.